Today we will be solving a question which came on GATE Xl-S 2018, and the question number here is question number 18, and this came from the topic of microbiology, and this came in the section of Xl-S. First, we will read the question. The question is stated as a continuous cell culture being carried out in a stirred tank reactor is described in terms of its cell mass concentration X and substrate concentration S. The concentration of the substrate in the sterile feed stream is SF, which is given as 10 gram per liter, and the yield coefficient, that is Y X divided by S, is equal to 0.5. The flow rates of the feed stream and the exit stream are equal. That is, F is equal to 5 ml per minute. And the constant and constant. They are constant and they are equal. If the specific growth rate is given by this equation, then the steady state concentration of S is. So this question, if you carefully read, so for solving this question, you need to have certain idea about the continuous cell culture conditions in a stirred tank. So now let us look into what it is. So it is known as the continuous flow stirred tank reactor, which is also known as in short form CFSTR. In biological or in microbiology, this kind of flow state tank reactor can be also called as a chemostat tank reactor. So what is this continuous flow stirred tank reactor? So this is one in which the contents are stirred so uniformly that it is assumed that no variation or concentration gradients exist within the vessel. So in the whole vessel, there should be no change or no variation in the concentration throughout the volume of this vessel. So this is when this kind of scenario happen. So this will be possible only when this continuous flow state tank reactor is present. It is actually possible theoretically, but somewhere it may also deviate, because theoretically it is ideal that it will be occurring like this, but there can be minute fluctuations which can happen. But what is important here to know that in a chemostat condition, what is important is that during the chemostat, the nutrients, the substrate or the nutrient which is limiting for the growth of that microbes in this tank reactor, that amount is actually uniform, or the rate of this change at steady state is constant, or the rate of the change is actually constant of that limiting reactant or limiting substance nutrient which is actually required for the growth of this bacteria in this tank. Okay, so there are certain parameters which this Q in, what is Q in, what is Q out, what is V, what is C. The C is the actually concentration. So the concentration will always be remaining constant. The rate of change of concentration of this will remain constant. So C out is equals to C in the tank. Okay, so there is different parts of this tank reactor. So one is this impeller which is connected to the motor, which will actually rotate and always keeping a aeration condition in this tank reactor, whereas there are different agitator, baffle, and there is a cooling jacket to maintain a temperature inside this flow state tank reactor. Now, what are the important things that you need to know for solving questions which come from this continuous flow state tank reactor? What is the flow rate? The flow rate of this kind of stirred tank reactor or chemostat reactor is given by this Q. The Q is equal to V by RT. So what is V? V is the volume of the tank or the volume of the tank reactor. And what is this R or this RT? This RT is the residence time. Okay, so how much amount of time your substrate is actually residing into the tank reactor, that is given by this residence time. So the flow rate Q in ml per minute is equal to the reactor volume, that is V in ml, divided by the residence time in minutes. It can be measured empirically in a reactor in steady state. Steady state means the rate of the change of your substrate at the steady state will be zero, as the volume of effluent from the reactor per unit of time. So in a steady state, if you take the volume of your effluent from the reactor per unit time, then also you can actually calculate this flow rate. In steady state, the inflow and the outflow of the reactor are equal, and the culture volume of the reactor does not change. So the net volume change is also zero in a steady state. Okay, the amount which is coming and amount which is leaving from this reactor, that is actually constant in a steady state. Then what is this retention time, or what is this residence time? The retention time is also sometimes called as this residence time. This is given by the formula V, which is the volume of the tank divided by the flow rate that we have just talked about. The residence time in minutes is the time it takes to entirely exchange the volume of the reactor, and is expressed as shown above. So entire volume of your reactor is getting exchanged. So everything is getting exchanged. So the time which is required for that exchange, the everything of your reactor to get exchanged, is your retention time, or the RT, which is given by this formula, that is volume of the reactor divided by the flow rate of the effluent which is leaving the system. Okay, and what is dilution rate? Dilution rate is given by Q by V, that is flow rate divided by volume. So that is just the reverse of the retention time. So 1 by retention time is your dilution rate, that you can also see from this formula. The dilution rate equals to the flow rate divided by the reactor volume. Now if you see that if your flow rate is faster or higher, then what will happen? Then the more and more amount of these nutrients will be leaving your reactor at a very fast rate, because the flow rate of leaving of the effluent is much faster. So as a result of that, your microbe which is growing in the state tank reactor, it will have less time for utilizing those nutrients, and more what will happen. So this, if your flow rate is actually higher, then your growth rate, or this growth rate will be slower, because the flow rate is higher. Now if the flow rate is slower, then the nutrients will be present in your solution and your growth rate will be higher. Okay, so from this, if you see that your flow rate is actually, if your flow rate is higher, then your growth rate is higher. If your flow rate is lower, then your growth rate is lower. So you can see that at this steady state, the diffusion rate and the instantaneous growth rate is equal. So it is very important for solving the question. At steady state, the diffusion rate, that is this D, and your instantaneous growth rate, that is U, is equal to this diffusion rate. So from this formula also you see that if you consider your volume of the tank reactor to be constant, that is actually present in the steady state, then since this D is equal to your instantaneous growth, so your instantaneous growth is directly proportional to the flow rate. So if flow rate is increasing, then your instantaneous growth will also increase, because the metabolic wastes are removed in a more faster rate, and more nutrients are actually added into your tank. But if your flow rate is slower, then this growth rate, the instantaneous growth rate, will also decrease, because a lot in a longer period of time, a lot of metabolic waste is getting settled down into the tank reactor. So that is why how this flow rate and this instantaneous growth rate depends upon one another. Now let us look back again to our question and what is being asked there. So the substrate concentration is mentioned here as S, and the cell mass concentration is mentioned here as X. The concentration of substrate in sterile feed stream. So it is a continuous cell culture that we have already talked about. they have given the substrate concentration that we need to find out in the steady state. They have given the equation of your growth rate, and also we know that this, if we actually somehow calculate this dilution rate, then we, because it is in a steady state, we can actually move directly to the growth rate, because at the steady state this diffusion rate and instantaneous growth rate are equal. So now let us, for this kind of question, one thing to note about is that you always have to look into the units. If there is some mismatch in your unit, then your whole answer will be wrong. So first what you need to do is that you convert all these values into the same unit. In means if it is in liter, it should be also in liter. Then only your answer will be correct. And since your in the question itself it is mentioned that they are asking the answer in gram per liter, so it is important if we can actually convert this milliliter into liter, so that our life becomes easy here. So how we are going to solve the question? The answer will be the mu is equals to this 0.3 s divided by 1 plus s, which is already given. So this mu is your growth rate, or the instantaneous growth rate or specific growth rate. Now this concentration of the substrate in the sterile feed is already given, that is 10, and the yield is given as 0.5. Now this flow rate is this F, which is mentioned by this 5 ml per minute. So here it is already given in your question that the flow rate F is 5 ml per minute. So what we have just converted it into liter per hour, because this is the SI unit. So we have just converted into liter per hour, and then the volume is already given. So what we need for finding the diffusion, the dilution rate, is your flow rate and the volume. The volume is already mentioned here as 3 liter. Just divide 0.3 by 3, you will get 0.1 hour inverse. So at steady state, as we have already discussed, that this dilution rate is actually equal to your growth rate. So just put the value here. Already this growth equation is given. So 0.3 s divided by 1 plus s. So the substrate concentration will come out to be 0.5 gram per liter. So what you can see here is that we have not used this SF and yield value. So these are some extra information that sometimes they are given in the question to confuse you. So you should be very clear about the reading the question and try to identify that what information is important for solving the question and what information you can neglect, so that you don't get confused in the examination hall. So this is the answer. The answer is 0.5 gram per liter. For your reference, you can actually go through these courses of NPTEL. So the first course is of Functional Genomics by Professor S. Ganesh of IIT Kanpur, and the second course, which is the Cell Culture Technologies, provided by or taught by Dr. Moinak Das of IIT Kanpur.